met before at some point. And I was going to say thank you for the bag of goodies in my room. I'll have to go on a diet, I think. Well, if you look in the brochure materials you have on your desk, there's a page in the back called Milestones in GCSF Development. I thought it's a useful place to hang tidbits about the history of how we got to where we are now with neutropenia and the registry and the NNN. But first of all, so many things happened after World War II or in the 50s in medical research. One of the biggest events there in terms of understanding white blood cells was the availability of radioisotopes to trace the formation of blood cells. And it was really from that research came the concept of stem cells, that there was a cell type that was the origin of all the cells that proliferated from it. And then another concept that was very important was that there are some cells that are made that live a long time or relatively long time in our body, and others that live a short time. And that the short living cells turned over or had to be replaced repeatedly and some daily. And it turned out that neutrophils are among the most rapidly turning over cells in our body and those that need to be replaced the most often. And that was really an important concept. The second concept that came as a foundation for us was how to grow blood cells. I remember this very vividly. I was working in a lab where people had just succeeded in growing lymphocytes. And then in Australia and in Israel, some investigators learned how to take bone marrow and grow those cells. And then from that research became what could stimulate those cells to grow. And a remarkable thing that was found is that in our bodies, there's some hormones that almost every cell can make, and others that don't do that at all. And it turned out that the hormones for the inflammatory response or for stimulating neutrophils to grow were made in all parts of our body. And one of the things that always interested me was one of the richest sources of what became known as GCSF was the uterus. And people have wondered why that would be. But also, in terms of daily regulation, the cells that are most important are the endothelial cells, the cells that line our blood vessels. It was in 1966 that the term colony stimulating factor was coined as a factor, a protein that could stimulate granulocytes or neutrophils to grow in a little petri dish. And that was really a remarkable observation made by Don Metcalf and others in Australia. And he coined that term, colony stimulating factor, which is stuck. And then if you know what happened in molecular biology is the purification of proteins became a rapidly developing field. And it was a German investigator, a very young person at the time, Carl Welty, close friend of mine, who purified GCSF and GM-CSF and a number of other growth factors at Sloan Kettering in New York in the research laboratory. And so the purification of a protein led another young investigator, Larry Souza at UCLA, to actually clone the gene, to identify the gene that made this protein, and then to introduce that gene into bacteria so that you could produce lots of it, enough for studies. And I knew Larry about the time that he had done this and became involved in the late 1980s with Larry and Don Metcalf and a number of other researchers in trying to find clinical applications. And I went to Amgen when it was a little company that people could have sat around the table here and said, I have this idea, would you possibly give me a little of this stuff to test? And I said, if you can show it works in cyclic neutropenia, it'll work for everything. And I was pretty grandiose and way beyond my knowledge at the point, but I was, I guess, a salesman. And they said, okay, and we began. And it was a good journey, and it's still going. But anytime you do anything like that, there's always some foundation. There's a lot of people who were involved in building up the case for why you would do something. Well, to compress what I have to say, look at this sheet and you can see the history and the flow of events. We did a study and there were other studies and then a randomized trial, and there was FDA approval of the drug. And I remember this vividly. It's kind of an interesting anecdote. I went to the FDA and testified about G-CSF, and they knew I was hiding something, but I wasn't. But anyway, it was sort of the how can we trust you experience. Maybe you've had that somewhere when you got a speeding ticket or something. Anyway, I also went to Europe, and same story, same data, and they said, Dr. Dale, we trust you. We don't need to have you say anything. And they approved the drug. Anyway, it's interesting the way that drug approval occurs, but the approval is important for us is that we conducted the study of using G-CSF to treat neutropenia. We couldn't put it into very neat boxes. And I remember saying to the people at Amgen who were the sponsor of the trials, they didn't want us to do anything that wouldn't work. And I said, well, how do you know it's not going to work unless you try it? And we couldn't distinguish the different types of neutropenia enough to know which ones would be best. So I said, let's go for the whole thing. And we did. And it turned out that, as I'll say in a few more minutes, G-CSF was a strong stimulus and worked for most forms of neutropenia. The registry started in '94 because the FDA and the European authorities, the EMA, required that we do a follow-up because they didn't know at that time if a growth factor would cause children to grow, be tall, 7 or 8 feet tall, or whatever else of their body would grow because they didn't know the target of the product for other cell types. It turned out to be quite specific. But anyway, we had to do actually 10 years of follow-up to prove that there was not some unexpected effect. The NNN group came after the wonderful research nurse who've worked with me, and many of you have talked to Audriana Boyard, and I thought it would be good to have a family meeting. We had several family meetings in Seattle, and then the NNN was formed with strong leadership from you and some folk who are no longer with us, but to codify and strengthen our relationship between families and patients. And it's been really a wonderful journey for me because I think it's been mutual. I've been interested, but you've been often carrying the ball in terms of treating yourself or your children. And we've tried to be as candid as we could with everything we know as soon as we know it. And we always know that, like in every other thing in life, there are questions. But that's been the wonderful part of NNN and the registry to make us all in the same boat. Now, Akiko, you're going to talk about now, right? Go to it.
Thank you, Dr. Dale. And I just want to do a big shout out to Dr. Dale. He really, I think, was visionary at the time that he set up the SCNIR to study this very rare disease. Other rare disease groups have turned to Dr. Dale, as have the NIH and other places, as sort of how do we organize in a coordinated way the study of a rare disease. And without this registry, really this type of knowledge would not have been possible. So I just want to give a huge shout out to Dr. Dale. And by way of introduction of myself, I am a physician scientist. I work a lot in the laboratory trying to understand the molecular mechanisms of these conditions to try to develop better diagnostics and therapeutics. And by chance, I ended up moving to Seattle. My husband, who's a cardiologist, was recruited to Seattle, so I sort of tagged along, and also started working in Seattle, where I had the chance to work with Dr. Dale, and he kindly sort of took me under his wing at that time. I worked with him in the SCNIR, and he helped us set up the Shwachman-Diamond syndrome registry. And I'm so happy that we've been able to continue this really productive partnership. Recently, though, we've continued the partnership across the continent, and the Center of Operations, for logistical reasons, have recently moved from Seattle to now Boston. As part of that, we continue, of course, to work closely together. But we've been able, as with any kind of move, been able to expand in different types of directions because each different academic environment offers different types of opportunities. So I'll share some of the new directions of the SCNIR with you, and also welcome your input. I hope you will all speak up here during the panel sessions, because we want to hear from you. What is important to you, and how can the SCNIR better partner with you and provide useful studies and information and resources for you? So when the SCNIR transferred to Boston, which was not a trivial thing, there's a lot of regulations that govern the safety of this type of research that involves partnerships with patients. And so we are required to reconsent everybody so that we can continue to study patients going forward. So if you were previously consented to the SCNIR, we can continue to study all your records that were collected in Seattle that are now transferred to Boston. But if we want to continue to study new events that are happening with you, or new patients, you have to reconsent. And so this is where Karen Brundage, who will be speaking with you shortly, has been leading our team to reconsent everybody. I would encourage everyone to do that. And also, please get the word out. I think there's, understandably, with any kind of change, sometimes some need for explanation. And we just need, in order to stay current with current regulations, reconsent people through the SCNIR, which is now based in Boston. But your data are not lost. It is continuing to be studied. So what are the SCNIR priorities? Well, our priority in the SCNIR is to really understand, to try to improve the health and quality of life of everybody, children through adulthood, with neutropenia. And we're focusing on how do we improve diagnosis, how do we improve therapeutics, how can we develop novel therapeutics, which Dr. Dale will talk about in a moment. GCSF has been a miracle in these very severe congenital, meaning starting at birth, neutropenias, which was previously had a very high mortality. Now it's been totally changed, but there are, of course, we can do better. So that's what we're continuing to work on, and we need better therapies for some of the other neutropenias. One of the major barriers to the study of neutropenia has been the natural siloing of medical care. There's pediatrics, and then you transition to adult clinics, and the two clinics don't talk together, right? It's quite separate, the medical systems. And so the SCNIR was a unique resource to really try to study across the age spectrum, across the lifespan. And we in Boston have made a big effort to develop sort of a joint pediatric adult program to offer both clinical care as well as to study neutropenia in collaboration between pediatricians and adult doctors. I didn't include their names or pictures here, but Dr. Nancy Berliner and Dr. Chris Riley are our adult partners, and we have this joint pediatric program with adults, and we have a joint clinical program where we talk together at conferences regularly and share our expertise. And I think that has provided some comfort to our families as their children grow up and graduate. They have a clinic to go to. And I'll talk about coordination of care if you come from elsewhere. Not every place has a center with a lot of expertise in neutropenia. It doesn't mean your doctors aren't expert doctors, but they might just need more data on neutropenia. And that's where the SCNIR can provide a resource for them and for you. Now, the registry is a partnership. So there are unfortunately no great animal models. There's no mice that conveniently model most of the neutropenia conditions. And so we really, in order to understand, and frankly, even animal models of diseases don't really model human disease that well. We've been able to cure lots of mice of all kinds of cancers, but sadly, those drugs haven't worked often in people. So you have to study people with neutropenia. People are not mice. And so this registry, started by David, has been instrumental in studying what happens to people with neutropenia by partnering with you. And I'll talk about that in a moment. One of the other priorities, which David very early on initiated, was, you'll notice the I in SCNIR. It's an international registry. There are certain novel barriers that are coming up right now in terms of what we are or aren't allowed to do in research, but we remain committed to collaborating across the world to share expertise in neutropenia, to share what we learn so that we all learn from each other. These are rare conditions, and if we don't share and learn from each other, we're not going to move forward fast enough. There is urgency. We don't want to solve things in another 50 years. We want to solve them right now for all of you so that all of you can benefit from this. And so we've developed many international projects. I've listed a couple here. You might have noticed Dr. Myers and I sort of convening before the conference. We're working very hard on completing an international study of Shwachman-Diamond syndrome, which is a particular type of neutropenia syndrome. But what we learn from that study can often inform other types of neutropenias. And this involves input from 14 countries around the world. Even though we have a large registry, we have one of the largest, we actually have the largest Shwachman-Diamond syndrome registry worldwide, but even that is not enough to answer some critical questions. And so now we have almost 900 patients pooled from around the world, where we're working together to understand what's happening with Shwachman-Diamond syndrome, what do we need to do to improve clinical care. So there's strength in partnership. I just wanted to introduce you to the Boston clinical team. Of course, Peter Neuberger and I work very closely together. So Peter has been an integral leader of the SCNIR together with David. He's been on the advisory board of the SCNIR for much longer than I've been part of it. And I've been very pleased to be able to partner with him. So when I left David in Seattle, I was able to join Peter in Boston to continue this work in the SCNIR. So all of the studies that I'm describing to you are certainly part of studies that are led by all of us. So our clinical research manager, I'm going to introduce you because if you contact the SCNIR, you may be interacting with some of our team. Leah Chung is our leader, and she organizes all of the SCNIR from a regulatory and organizational standpoint. Karen Brundage is our research nurse. She's also a highly experienced NP on the clinical side with a treasure trove of experience and has been a wonderful addition. And she used to work with Adriana actually back in Seattle when she was in Seattle. You'll notice a little theme here. And that we continue to work closely, of course, with Adriana. And then Edie Weller is our biostatistician. You'll never meet her, but I just put her here because she really keeps all the studies at a high level of rigor. You need high scientific rigor and the best statistics to make sure that what we're saying is appropriate. And then Liz Korn is a recent clinical research assistant who does most of our outreach directly to you. Now, what is a registry? Now, I feel like in a way it's unfortunate that the term registry was chosen, although I understand why, because in this day and age, registry sort of connotes a place where people are registered, right? So we just sort of stick your name on this registry and that's that. But it's actually really a partnership. It's a study, ongoing study that's a partnership with all of you. And why do we need this? Well, The medical records contain a treasure trove of data about each person with neutropenia, right? There's so much data there that we all need to learn from, but unfortunately, they're all locked away, right? In individual hospitals and individual clinics. Nobody can really access that to study it. So the registry, and in addition to the medical records, there are clinical samples, which, after your clinical tests are done, the samples are thrown away and discarded. And this is very precious material that we can study to learn from. So we're trying to now, through the registry, gather this together so we can study it. It's kept confidential. Confidentiality is very important. We have to follow regulations that govern the safety of this research, and it allows us to then collaborate. So it's not sort of a black hole where all the data come. We can then share that in a sort of de-identified, confidential way with other researchers so that everybody can learn as much as possible. When the samples are sent, so a little bit of logistics, we can send out, for those of you who aren't familiar with the SCNIR, if you consent, and that you have to give informed consent for this, whenever you're having samples, a bone marrow sample drawn, and at that time we often will collect a blood sample, or if you're not having a bone marrow, even just a blood sample drawn as part of clinical care, so there's no extra needle pokes. If you let us know, we can ship you a mailer. Or if you don't let us know ahead of time, we can send by email a little mailer sticker to your doctor, which is prepaid. And they can ship the samples that are being drawn. They can just draw a little bit of extra and mail it to the SCNIR so we can bank it and study it. These are very precious samples without which we cannot do the types of research that I'm about to discuss. Just a little caveat, if at all possible, and you're going to do this, and we're very grateful if you do, don't have your samples drawn on a Friday. If it's clinically urgent that it be drawn on a Friday, go ahead and draw it by all means. But the samples sit over the weekend. I don't have a way to access them over the weekend. They sit in the mailroom of the hospital. And the cells get old and they're harder to study. So if at all possible, send earlier in the week. So what are some of the current projects that we have ongoing? So I'll just describe a few of them at a very high level, and then we can talk some more during the Q&A session, and we would love to hear from you what's important to you. So we're trying to understand more about the genetics of neutropenia. There's a whole talk on this that I'll give later, but if we can pinpoint the reasons for the neutropenia, and in many of these cases, there are genes that are not functioning correctly that can contribute to the neutropenia, that can give us a clue about how to better target therapies, and I'll give you some examples later in the afternoon. We're working very closely with colleagues over at the Broad Institute. Some of you may know the Broad Institute. That has been a leader in this field of genomics, and we can take advantage of their cutting-edge technologies that they're developing. And we've discovered we have some new data coming out, and I'll share some of that with you later in the day. We're also working hard on the immunology of neutropenia. What is autoimmune neutropenia? Can we understand the causes better? Can we come up with better diagnostics? As Dr. Neuberger just mentioned, the anti-neutrophil antibody test is not very good. A positive doesn't mean there is an antibody there. A negative doesn't mean there is no antibody there. So it's not a very good test. We need better tests. We're working very hard with colleagues, experts in immunology, to study these samples, very precious samples, to try to understand this and understand, can we predict, for example, currently, if a little child comes in, as Dr. Neuberger said, with what we think is autoimmune neutropenia, and we think it might get better, we have to follow that child very closely with regular blood counts to make sure that they get better. Wouldn't it be nice if we had a test early on that told us this child is likely to get better? You don't need to keep poking them for blood so often, and you can just monitor them, more safely. So we're trying to come up with, and also it relieves a lot of the stress of coming into the ER for fevers all the time. So we really want to try to improve that. Gene therapy, there's a whole talk by Dr. Neuberger later on gene therapy, but I'm just going to give you a high-level overview. The goal of gene therapy is to take the patient's very own blood cells that are currently having trouble generating neutrophils and fix the gene that isn't functioning quite right, and then give those cells, the patient's very own now repaired cells, bone marrow cells, back to the patient so that now the patient can start to produce neutrophils normally. And we chose as our, we recruited, so part of what we do in the SCNIR is recruit sort of the top thought leaders in the field and convince them to work on neutropenia. And Dr. Bauer's lab happens to be right next to mine over at Boston Children's Hospital. And he, as Peter said, there's been a lot of talk about gene therapy. Dr. Bauer is one of those people who not only talk the talk, he walks the walk. He has completed successful gene therapy trials with a team on sickle cell. You may have read about this in the New York Times, where we can cure sickle cell disease with gene therapy. He's also been involved in some of the trials with adrenoleukodystrophy, which have been very successful with Christy Duncan. Peter will talk about the Elaine neutropenia studies. I'm pleased to say that the Schwachman-Diamond gene therapy studies are looking great. And if we can get funding, we hope to open a clinical trial in the next 2 years or so. The science is not the barrier there. It's really the money. There's no money. So we're trying to get something going there. But the science has been great. So we hope that there will be more hope. It would be nice to be able to cure the neutropenia in that regard. One of the things that many of you have to deal with some of these neutropenia conditions is a potential risk of leukemia, right? And in order to try to identify situations where leukemia is imminent, the recommendation is to monitor the bone marrow regularly. But this is painful, stressful. For children, it involves anesthesia. For adults, they just don't want to do it often because, who has time? They're in college, they are working. So we're trying to develop blood-based surveillance strategies. It will not be perfect. The bone marrow is the best, but wouldn't it be nice if we had a screen to let us understand, well, these patients might be most at risk. These are patients who really need a marrow, whereas other patients we might be able to monitor safely just with blood. We need to understand this a lot more, and it's specific to each type of neutropenia, but we've made a lot of progress in Schwachman-Diamond syndrome, and we hope to have more to share on that in the next few months. So we're really working hard in collaboration with Coleman Lindsley over at Dana-Farber, and Felicia Andreasen is a postdoctoral fellow in my lab, working on sort of blood-based surveillance strategies to look for leukemia risk. And then this is a current project that we're very excited about that just recently opened, which is currently what we understand about neutropenia is from the doctor's perspective, right? There are publications of doctors' reports of the patient's experience of neutropenia, but it's different, right? If you're actually the patient or the family experiencing neutropenia. So this is an effort to hear directly from the patients and families and hear your voice. This is called a patient-reported outcome study. That's just the technical name for the study, but it involves a survey. This is a validated survey that has been validated to meet certain metrics for this type of analysis and this type of study. And we've recruited experts. Dr. Stacy Arnold is well known for her work in sickle cell disease, and she's now recruited her into studying the neutropenia. And she's working with Tom Mishniaki, who is a trainee of Dr. Boxer. Many of you know Dr. Boxer. And basically, it involves about a 15 to 30-minute survey online. If you're on the SCNIR, you should have received an email about this. We want to hear from everyone, adults, children, parents. And the issue is some of you will have received a secure email that requires that you check a link, click a link, because we have to have your permission to send a non-secure email. If it's secure, everything is encrypted and you have to click the link. And if you're like me, if I see anything like that, I think it's spam and I don't open it, right? Because, maybe it's a malicious thing. So if you have any questions about that, the title line is Share Your Neutropenia Experience with Us, but that sounds a little scary. So we're changing it to Neutropenia Update from Boston Children's Hospital. It will have a link. If you want to make absolutely sure that it's really from us, you can call the Neutropenia Registry and say, hey, did you just send this email? I just want to make sure with you before I click on it. But this will stay open for just a few months, and it does, just in case your kids are interested or you're interested, there is a little gift card that you get from Amazon if you complete your survey. So just a little carrot. So thanks to everybody for participating in the Neutropenia Registry. Some of you ask, how can I help? And so for that reason, I just wanted to mention that there's a little button on the Neutropenia website, just because many people ask if people want to donate to the registry. So thank you so much.
If I can just add, this study of patient and family experience is open to people who are registered with the SCNIR, but if you're not registered and you want to participate, just contact us and we can register you and get you your voice heard on this survey.
I just wanted to add that Karen did bring consent forms if anybody is interested. Sometimes, once you get home, you get back to life and it's distracting. So Karen brought consent forms if people want to join the registry and haven't had a chance to do that yet. So while you're here, you will have that opportunity.